Great, thanks. And much appreciate the invitation. Looks like you have my deck. All right, now can I advance the slides? Is that? Oh, okay, easier. Great, thanks. So what I'm going to try to do is give you a bit of a view of, at least from the FDA perspective, of some of the changes in the types of drugs being developed. Some of this may be a bit carrying Cole's Newcastle, you may be well aware of some of these changes. I do want to talk about, since we're seeing the shift towards more rare disease development, talk a little bit about some thoughts and some challenges in developing drugs for rare diseases, as well as in the current context, drugs for more common diseases. I do want to talk a little bit about just what the approval standards are, but also how one can think about how one has to adapt them to changing types of applications. And then also talk a little bit more about some of the changes in the way clinical trial programs are being developed. Some of the changes that I think have been going on for actually decades, but certainly accelerated over the past years. So when we think about some of the changes in sort of the landscape, both the regulatory as clinical and a scientific background, we have been seeing the shift towards more applications for rare diseases but also for narrower subpopulation. That's particularly in oncology, but it's not just oncology. There's a lot of reasons for this. Again, I'm guessing that you're familiar with it. Obviously, the opportunities in more common diseases, in contrast to 20, 25 years ago when I first joined the pharmaceutical industry, now there are multiple approved drugs for many common diseases. When I joined industry in 2000, there were very few drugs for type 2 diabetes, and now there are many, many classes for type 2 diabetes. It doesn't mean that there's not unmet needs, but certainly the opportunities have narrowed, and the need for differentiated therapies that really offer distinct advantages has risen dramatically. There are, of course, many rare diseases, a vast number of them, and many or most of them still have unmet needs, despite a lot of progress in rare disease drug approvals. The science is evolving. I mean, that's led to new platforms, which we'll talk about, but obviously genomic drivers, molecular drivers that have made huge differences in oncology, but also understanding the genetics, which has made a difference in areas like cystic fibrosis, and led to new focused targeted therapies. Obviously, the Orphan Drug Act has led to economic incentives for rare disease drug development, but obviously there are other factors related to the economics of drug and markets, which are well beyond my remit and knowledge. And of course, as we've understood common diseases, we've been able to divide some of these into relevant subpopulations, and that has led to molecular targeting for those as well. Now, as I said before, there isn't dramatic opportunities, I think, still in common diseases. We've seen this with shifts in the treatment for type 2 diabetes, heart failure, obesity has been dramatic. We just approved another very effective drug for obesity yesterday, and of course, drugs for Alzheimer's disease. So there still is huge opportunities in common broad treatments for common diseases, but those opportunities require truly differentiated therapies, often that really are leveraging our understanding of the background disease pathogenesis and the utilization of new platforms as well. So in terms of some of the changes in how we approach drug development and regulation, I'm going to talk a little bit more about the approaches in rare disease drug development and how we think about the regulatory environment there. New approaches to evidence generation, which is important because it changes potentially some of the costs for doing development, some of the approaches, some of the types of evidence that are generated. Obviously, over the years, including with the recent legislation with GDORA, an increasing focus on diversity in trials. I'll say a word or two about that. And I think as a theme we've seen over the past decade, the importance of the patient really at the center of what we all do in terms of thinking about making sure that the drugs that we're developing are targeting the burdens that patients are facing, but also the way we design trials and the endpoints we use really are relevant to patients. We put out 4 guidances relevant to patient-focused drug development, focused on endpoints and PROs. I won't say more about that, but it's been a clear theme and an evolution over the past decade. So saying more about drugs for rare diseases in terms of the development and regulatory approaches to these, of course, there has been a lot of interest in using, rather than our sort of standard two adequate, well-controlled trials to establish substantial evidence of effectiveness. I'll come back to all of this a little bit later. There's been an increasing interest in confirmatory evidence, and that requires a greater investment from companies in understanding the molecular background and the pathogenesis of disease, and performing more extensive work earlier on, both in terms of animal model work as well as human translational work, which companies are not really doing, I think, to the extent that needs to be done. There's more interest in surrogate endpoints or ways of trying to get drugs approved more rapidly without the investment that many companies need to make in developing the rationale, the background evidence, and the support for those surrogate endpoints. There's certainly more interest in accelerated approval as a more expedited pathway to get drugs approved, with, once again, not always the background support that's needed to meet the standards for accelerated approval, which are still high. The issue of regulatory flexibility, of course, comes up with rare diseases. I'll talk more in a little bit about what we mean by regulatory flexibility, as well as what we don't mean by regulatory flexibility. And certainly there has been a lot of innovation in the approaches and designs and analytic methods for clinical trials. We have programs that look at complex innovative designs. We have model-informed drug development programs at FDA, all intended to try to find efficiencies in how data can be reliable and generated more efficiently. Another unique challenge over the past years, again, not new, but really coming to the fore, is what one might call N1 programs. And I'll say a few words about that towards the end. There's also a whole range of new approaches to evidence generation. So I'm going to say a little bit more about that now and then come back to this and talk about them in some detail. So as I said, there's certainly been a push over the past years, and our current commissioner is one of those pushing, about new ways of evidence generation. And he has had a long career in trying to push towards randomized trials that are more pragmatic, generating much evidence from larger programs, which really does make a great deal of sense. And there's many tools that have evolved, and these are not new. Decentralized trials go back 20 years and more, but there's been a lot more focus on decentralized trials over the past years, and new tools that facilitate decentralized trials. The focus on pragmatic trials, again, this is not new. We've been talking about this for decades, but more focus on this, including more focus on point-of-care trials, something that came up during the COVID pandemic. The use of master protocols, once again, certainly not a new concept, but certainly accelerated over the last years. I'll say a few more words about these in just a moment. Digital health technology endpoints. There has been a dramatic evolution. Again, there has been efforts in this area for many, many years, but with many more tools, and of course, with us all carrying our own digital devices, the opportunities for collecting information from patients in the real world, as it were, in their homes, in their workplaces, to understand how drugs might improve their outcome is really at the forefront. And I think we're just beginning to see how this might be used, as well as some of the limitations. And then finally, the use of real-world evidence to support regulatory decisions. As I'll come back to, real-world evidence has been used for decades in regulatory decision-making, but the opportunities to enhance the ways it's used, to increase the robustness of the data, give us the potential to use these kinds of data more and more for regulatory decisions related to labeling and effectiveness, as opposed to simply for safety. Now, I mentioned before something that, again, I think is probably very well understood by all listening, is that there has been this move towards more and more applications for rare diseases. This is just looking at the number of NMEs over the past 12 years or so. The pattern for 23 hasn't really changed. As you can see, going back about 12 years ago, when first we looked at this in 2010, the proportion was 25 to 30% or so of applications were for rare diseases. And now we're moving more and more towards seeing about half of our applications for often designated for rare diseases. I don't know that the number is going to go beyond that, but certainly I don't see any expectation that that number will go down. Indeed, if you look at active INDs in rare disease, the number has year on year increased. This just goes to 21, but it's continuing to see the same trend. Subsequently, more and more applications for rare disease INDs, more work in this area, and I think ultimately that will continue to mean that we're going to see more continuing high proportion of NME, NDA, BLAs that are for rare diseases. Now, rare diseases obviously face a number of challenges. I'm sort of listed on the left some of the things that are well established and make common disease drug development relatively easier. Large populations, so recruitment usually isn't that much of a challenge. Easy for me to say at this point. I spent 20 years being challenged by recruitment issues, but standard approach of getting to 2 adequately well-controlled trials, fairly well established. It was obviously a wide range of disorders that one targeted, well understood natural history, guidances and precedents from FDA and from other regulatory agencies about how these drugs should be developed, and endpoints that were well accepted, LDL, blood pressure, A1C for type 2 diabetes, et cetera, that allowed for relatively clarity in terms of how one develops and designs trials to support these diseases. Now, in contrast, in rare disease development, we're facing these small populations, varying sizes. Sometimes these are thousands of patients, but sometimes we're talking about 50 or 100 patients in the US and double that in the global population. It requires novel study designs, which we'll come back to. And these are typically progressive, serious, life-limiting and life-threatening diseases, and they lack approved therapy. So there's really a need for these treatments. There's a lack of precedent for drug development. What are the endpoints? What are the trial designs? What are the inclusion-exclusion criteria? They're not cut and paste from prior work. That means that they have to be thought through very carefully to make sure the trials are properly designed. There's a lot of diversity of these diseases. It's not just a single presentation. There's both genetic and phenotypic diversity. Despite similar genetics, the presentations can be quite different, and many variants of the disease exist. And that leads to a natural history, which also can be very variable and is often not well known, which makes trial design very challenging. How long should the trial be? Well, it depends upon what the rate of progression of the disease is. And if you don't know the rate of progression of the disease, designing the trial can be a bit of a fool's errand, and it can lead to trials that lean in one direction to suggest the drug might be doing something, but don't meet the standard statistical requirements to meet evidentiary standards. Drug development tools are not typically readily available across all of these types of diseases. We're working very hard to try to encourage and work with sponsors to develop these tools, but it makes it more challenging when you're entering an area trying to find a treatment for disease for which there's really no established endpoint. So we have a program that it fosters the development of such tools and endpoints. It was part of our last PDUFA cycle that we proposed, and I think that will help to develop types of endpoints that may have broader use across other diseases rather than just the target disease that particular program is focused on. There's, of course, ethical considerations. Many of these diseases are children, and the kinds of trials that can be done in children obviously are different. There are different regulations that we have to consider. I do want to just mention that we launched now a bit more than a year ago what we call the ARC program, Accelerating Rare Disease Cures Program. And the vision of this is really to increase and speed the development of effective and safe treatments for individuals with rare disease. And this is a sort of a multidisciplinary, multi-office and indeed center effort. It's within CEDAR, where it would include our Office of Translational Science. It includes my office, the Office of New Drugs. It's like we bumped one, sorry. And a number of other disciplines coming together to try to work with sponsors, work with the stakeholder community, work with the academic community to find ways we can accelerate drug development for rare diseases. I do want to just touch on issues related to common chronic diseases. Even as we more focus on rare diseases, there's still a lot of opportunities, and many diseases are still quite inadequately treated. So there's really opportunities for finding new drugs that are effective in common diseases. I spent 20 years doing drug development in diabetes, and I really thought when I left there was not a lot of opportunity. And as I left, we see the more and more information about the value of SGLT2 inhibitors, of GLP-1 now, of GLP-1 GIP drugs, which are effective also have been shown to have cardiovascular benefit. And so we're seeing a whole range of different effects in diabetes, shifting the focus of how we treat that disease. In obesity, I worked on drugs for obesity for many years, and the history there was not exactly a very favorable one, with drugs entering the market and then having to be withdrawn, such as sibutramine or lopinavir, for various safety issues. And only recently have we seen drugs that are quite effective for obesity, and I think we can expect to see more drugs for obesity. Obviously, this is a challenge in many ways, economically and otherwise, policy-wise. But from our perspective, seeing effective drugs in this area is really something quite remarkable. Drugs that are for congestive heart failure, Alzheimer's disease has been an area of development, and there's many different targets there. More recently, of course, the approval of drugs targeting these amyloid plaques, but I think that's just the beginning of a field that is going to see a number of novel therapies that are increasingly effective. But of course, we're also seeing in common diseases a focus on subpopulations of patients. And this really goes hand in hand with a better molecular understanding of the disease pathogenesis, being able to subtype diseases. This has just led to the incredible advances in oncology focused on molecular drivers and subtyping diseases that were previously seen as large categories, like breast cancer or non-small cell lung cancer, or varieties of lymphoma, where we now understand within subtypes molecular drivers and can approve drugs specific to those drivers with dramatic efficacy, even curative efficacy for some of these drugs. As with subtypes, we've seen advances. I mentioned cystic fibrosis, not more of a rare disease, but again, illustrating that defining the subtypes, defining the genetics or the genomics really can lead to dramatic advances in disease. It ultimately can lead to changes in the nosology of disease. I mean, diseases were defined by their phenomenon, by the phenotype. But if we understand subtypes, we might start, there has been proposals to talk about type 3, type 4, type 5 diabetes. I'm not aligned with that, but I think it illustrates the fact that as you understand the differences then in the types of categories of patients that might have different pathogenic mechanisms, drugs can be targeted more specifically for those subtypes. On the other hand, we also see a lot of proposals for subtypes that are not really subtypes. A subtype doesn't mean that someone has a slightly greater or slightly lesser efficacy of a drug. It means that there's really a different pathogenesis, maybe a different molecular driver for the disease, and really a targeted disease is only a targeted drug is only going to be effective in that subtype. So there's work to be done there. So I think that is an area that we'll see marked growth in over the years to come. Of course, we're seeing disease more and more treatments for resistant disease. We've seen this in depression, some really remarkable advances there, and in late stages of disease. So, narrower focus, but still important aspects of disease treatment. And of course, fundamentally, we're trying to find the molecular underpinnings of these diseases that we can target and ultimately lead to cures. And there's many chronic diseases where we are not there yet. I do want to just sort of, as I sort of launch to talk more about some of the new methods of evidence generation, it's hard to not give credit in a way to the impact of COVID. I mean, I think someone said never miss the opportunities of a good crisis. Well, COVID was a horrendous impact on all of us in terms of the mortality, the morbidity. I think we all had relatives, friends, colleagues that we lost who ended up terribly ill in the hospital. So the human toll was remarkable, but at the same time, it really changed how we had to think about working. We all, I'm sure, many of us probably work more at home than we ever did. I never worked a day of my life at home, and now I work 4 out of 5 days at home. So, our lives have changed. But it certainly changed how we think about drug development. It really accelerated our thinking about decentralized trials because, of course, patients often could not come to the investigational site. The use of DHTs, which enables decentralized trials, became increasingly important. How can we avoid having to have the patient come in to collect an endpoint? Could that be done by collecting data from the patient while they're at home? And DHTs fit perfectly into that need. Master protocols, will come back to that in a moment. Something that, again, has been going on for decades, were markedly accelerated during COVID because it was a way of really testing multiple different therapies concurrently. Large pragmatic trials, recovery trial in the UK led to identification of dexamethasone steroids as an effective treatment for in-hospital patients. Remarkably, within I think about a year of the epidemic, that data came out because of the efficiency of that approach to developing evidence. And it also underlined the need for innovative approaches, analytic approaches that allowed for a really sort of fit for purpose approach to how we would understand and design trials. And it also, I think, shined light on opportunities for real world evidence informing regulatory decisions, not so much necessarily informing approval decisions, but certainly post-approval, giving us more and more information about effectiveness. It also gave us tremendous information about the ways the disease were presenting, the impact of the disease, how it was being treated. So it was an incredibly informative source of information through the crisis. So I want to turn to talking about pathways and requirements for drug approval. And again, a lot of this may be very familiar to you, but I want to talk about it in terms of how one fits to the current needs as we move towards more subtypes of disease and rare disease approvals. I must say, despite working in industry for 20 years doing drug development, I had never really read the statute that I was designing trials to address. So I think a lot of people don't actually go back to it. But what we require to show that a drug works, to approve a drug, to achieve what we call substantial evidence of effectiveness, is really defined in statute. And it says what that is evidence consisting of adequate and well-controlled investigations, plural, which is where we get two trials, including clinical investigations by experts qualified by scientific training and experience, which is us, to evaluate the effectiveness on the basis of which it would be fairly and responsibly be concluded that the drug will have the effect it purports was represented to have. Well, putting all that together, we often require two active, well-controlled trials with p-values less than 0.05. But one thing I often point out is that there's no p-values in the statute. P-values are the way we interpret the statute in providing us with the persuasive evidence that's needed. And our requirement for these two active, well-controlled trials is really a scientific standard. Replication of an experiment is the way one can really be sure that the results are reliable. I do want to just mention, and I flagged this a little bit earlier, that there are three different kind of approaches, if you will, to meeting the effectiveness standard, the substantial evidence of effectiveness. The standard approach has been two active and well-controlled trials, which is standard, particularly because many of the programs that we were reviewing 20 years ago, 30 years ago, were for common chronic diseases, where this was a very feasible and appropriate approach to developing the evidence necessary to meet substantial evidence. But also in statute, came in 1997, was the ability for FDA to agree to the use of one active well-controlled trial, along with what was referred to as confirmatory evidence, which is a wide range of possible sources of evidence that supported the fact that the drug works. And we just put out a guidance on confirmatory evidence literally within the last months that explicates the type of evidence and the type of the sources of evidence that can be used for confirmatory evidence. Now, less commonly, we also accept a single trial, but that really is not a separate pathway. We think about that as a single trial that provides the same level of evidence as two active, well-controlled trials. So this is typically a very large, an 8,000 or 10,000 patient study, many sites, many different endpoints that provide similar confidence and persuasiveness as two adequate, well-controlled trials. Now, regulatory flexibility is actually embedded in regulation. It's in several different parts. Just to read some of the key words, while the statutory standards of safety and effectiveness apply to all drugs, so you still need substantial evidence, but the kinds of drugs that are subject to them and the wide range of uses demand flexibility. And FDA has determined that it's appropriate to exercise the broadest flexibility in applying the statutory standard. So what do we mean by that? Well, we can think about that in sort of two different ways. One is there are a lot of tools we have that are within the statute or within our regulatory framework that can be applied in rare diseases, accelerated approval, the use of one active well-controlled trial, and confirmatory evidence. These are tools we have to apply regulatory flexibility. There's also study designs that we can use that apply regulatory flexibility. The use of external controls, for example, rather than a randomized double-blind placebo-controlled trial. So those are approaches to regulatory flexibility, if you will. But the other component of that is the acceptance of uncertainty. When you're dealing with a population of 75 kids with a rare, serious disease, we can't expect two adequate, well-controlled trials. We have to show flexibility, and the acceptance of some greater level of uncertainty is part of that concept of regulatory flexibility. But in all cases, we still have to meet the standard of substantial evidence, which means we have to be persuaded that there is evidence that the drug has the effect it purports to have. Now, I do want to just sort of remind you that there's sort of two components, at least the way I kind of frame it, there's two components to how we approve a drug. One is what I was just talking about, substantial evidence of effectiveness, the evidence, the standard. And the other is the benefit risk approach. We think of a drug as safe if the benefit exceeds the risk. Seems kind of straightforward. Every drug has a risk. So how much risk is acceptable? How much risk would allow us to still conclude the drug is safe? Well, if the benefit exceeds that amount of risk, then that would meet that standard. We have a guidance that discusses it. But that's a pretty hard calculus, I'll say. I'm not going to, I think it's a discussion we could have several hours of time on, but I won't say more other than that's how we would conclude a drug is safe. Just to remind you of the approval pathways, the traditional approval using an endpoint that reflects how patients feel, function, or survive, and the accelerated approval pathway, which uses a surrogate that's reasonably likely to predict benefit. So we accept uncertainty by doing that, and that, of course, is in statute as one of our tools. So I want to talk a little bit now in the last 5 or 10 minutes about some of the changing face of clinical trials, because I think the way we generate evidence is evolving, and it's going to evolve dramatically over the next 10 or 15 years, I have no doubt. I'll talk about some of these, not all of them, but some of them I just want to say a few things about. But I want to start by just saying that while the way clinical trials, the clinical trial enterprise will be changing, and I think in dramatic ways, the standards are not going to be changing. So substantial evidence and benefit exceeding risk are going to be fundamental and unchanging expectations, even as the way evidence is generated may change. So a few words on decentralized clinical trials, because these, I think, will evolve. I don't think they're going to take over in the traditional site-based trials, but we will see more and more of decentralized trials. This is with some or even all of the visits done remotely, video conference type visits. Investigators will then potentially be sort of at a central location working with local investigators, drugs being sent directly to either local depots or directly to the patients. And it's going to be useful for a wide range of trials, pre-approval and post-approval, but not likely trials where drugs have significant safety concerns or where administration processes are complex. Digital health technologies, I said a few words about earlier. Of course, we're all carrying them, which makes this a lower hurdle. And there's a wide range of possible uses that can enhance clinical trials. In simple forms, things like adherence can be monitored, but more impactfully, novel trial endpoints, collecting patient-reported endpoints in an easier way, certainly something that's been done for years with these, but actively assessing endpoints, looking at time tasks or looking at passive collection of data from patients' ambulation or a variety of other things and activities that patients involved with becoming themselves endpoints in trials. The ways we can do safety monitoring, looking at blood pressure, looking at other physiologic parameters to measure safety. And of course, in the simple ways, looking then to connect and engage patients with clinical research, and of course, a wide range of sources of this digital information. I think you guys are quite familiar with the types of digital health technologies that can be turned into and used to support endpoints for clinical trials or for other clinical trial applications. A few words on pragmatic randomized clinical trials. There's been this sort of contrast between traditional versus pragmatic trials. The concept was that traditional trials were very strict, very narrow, did only look at a very small sort of subset of patients, and therefore were not generalizable. And that was in contrast to pragmatic trials. Frankly, that's a dichotomy I don't agree with. I think more and more our phase 3 trials have broader inclusion criteria. We're certainly pushing sponsors to try to ensure that they're as broad as possible. And so I don't think we have a situation where they're so narrow, but certainly the traditional phase 3 trials were often dedicated research sites, had more strict entry criteria, blinded and randomized and strictly monitored. The concept of a pragmatic trial is a much more open entry criteria. The practice sites becoming the point of care and the point of the investigational site, open label more often than double blind and still randomized, and using many fewer visits and central monitoring rather than the traditional site monitoring visits. Of course, increasing clinical trial diversity is one of the potential advantages of this. The use of decentralized trials at point of care and use of real-world data elements all sort of fostering this progression to more pragmatic trials. And as I said, I tend to think that even the trials that we now have industry running in our phase 3 programs tend to have pragmatic elements, be more open, be simpler types of trials. But we need to push on that to make sure that the evidence we get is more generalizable. Turning to real-world data and real-world evidence, again, these have been exciting times with a lot of progression over simply the time that I've been at FDA, which is now about 7 years. Real world data, of course, is the data that comes from a whole range of different sources, electronic health records, a lot from claims and billing data, but also from product and disease registries, patient-generated data from smartphones and apps, and so a variety of different sources. But what real world evidence is taking that data and being able to take meaningful information from it that may support regulatory decision-making. So it's really generating from the data evidence of the drug's effect. The uses of real-world evidence traditionally have been focused on pharmacovigilance, safety assessment, satisfying post-approval requirements, but extensive experience in rare disease has also been developed over the last 20 years with externally controlled trials. There's, in the simplest form, the value in using these data sets to identify a trial population. But as we learn more about this and as improved techniques and methods have evolved, there's also the potential for the use of this type of data for confirmatory evidence, but even the ability for this data to be considered an adequate and well-controlled study to support the substantial evidence of effectiveness. And we have approved a drug now, program, tacrolimus, for a specific use using a data set from a registry that is essentially real-world evidence. There's a lot of reasons why this is important. Of course, it gives the opportunity to take information from a much broader and diverse patient experience. The potential, of course, for really massive, sample sizes, tens and hundreds of thousands of patients, and of course, a lot lower resource intensity. If you can download a large observational database, develop a protocol to figure out how to probe and analyze it, you can potentially get relevant data in a matter of months rather than a matter of years at a cost that is dramatically lower. Just to say that there's really a wide spectrum of uses of real-world data and real-world evidence. Starting from the left, for more within the context of a more traditional randomized trial, we can start looking at, in simple terms, the use of real-world data to identify patients and to look at the feasibility of a trial to be conducted. We can take endpoints that are complementing the traditional endpoints. In the middle, there are trials that can be entirely based upon real-world evidence. So pragmatic randomized trials, for example, the endpoint might be coming out of claims data or might be coming out of EHR data. And then, of course, to the right of this are observational studies where all of the data is from healthcare interactions analyzed in a prospectively defined analytic strategy. I'll just say a few things about master protocols. Again, this is an area which has also seen a lot of growth. This is the concept of being able to test multiple different drugs across disease or disease subtypes and leverage a lot of efficiencies, common screening and enrollment procedures, governance, operational systems. And I'm not going to go through the different types of master protocols. The one on the bottom is perhaps the most relevant. This is the one that was really utilized extremely widely during COVID and led to a number of important observations. And the idea is you could start with evaluating a couple of drugs. A drug that didn't work could be dropped out, another candidate could be dropped in, allowing for a very flexible approach, more efficient than starting new trials each time you want to evaluate a different drug. Well, I want to end on 2 points. One is on this N of 1 concept, and just to introduce this, to present a patient to you. This was a patient that was reported in the New England Journal, a 6-year-old girl with a really horrendous progressive neurologic disease with a seizure disorder. It's called Batten's disease, which is an autosomal recessive single gene disorder. And she had a particular variant. There was a antisense oligonucleotide that was developed that was intended to sort of bypass this variant and normalize this defect. On the bottom, in fibroblasts, you could see improvement in the fibroblast on the left. Vacuoles that reflected the disease pathogenesis improved with the treatment of this particular antisense oligonucleotide. And sort of in an N of 1 fashion, this investigator from Mass General showed that there was a suggestion that the seizure frequency, which is what the graph is illustrating, going from left to right, reduced. Very hard to interpret N of 1 trials, of course, but obviously somewhat promising information. But the challenges of this have been multiple. I mean, what type of evidence do we need before you can expose a human to a drug? How persuasive should the mechanistic or functional data be to permit a clinical study? How do you figure out what dose to use or what regimen should you use? How well should product quality be characterized? You're using this drug to treat a single person. And how do we consider the patient situation, the rate of progression and the seriousness of the disease? How do you evaluate efficacy and how do you evaluate safety? And fundamentally, how do you take a drug of this sort that might treat 1 or 2 patients and think about how that fits in with our model of approving and marketing a drug? I think many of these questions we don't have yet answers to. We put out 4 guidances on this, but I think this is an area that continues to challenge both in the development area and in regulation. And I'll end just on a note about diversity. This has been a key issue for us over many, many years. Increasingly targeted through Fedora with the legislation that instantiated diversity action plans that need to now be submitted with drug development plans. And there's many reasons why this is important. Obviously, we want to understand the differences across different populations in drug response. We want to make sure that there's as broad an experience as possible for safety. We want patients to recognize that the drug has been studied in patients like them so that they feel like they can believe that the data generated was relevant to them. And obviously, there's an equity issue in terms of access to trials across different populations. Well, I've gone through a lot. I'll stop there. Happy to answer questions, but appreciate your attention.